Good morning, everyone. My name is Brice LeFrancois. I'm a senior R&D manager on the technology team, overseeing some of the research effort both at Diversigen and DNA Genotek. And today my talk is going to be about multi-omic visualization of the human gut microbiome. And I think the past 10, 15 years in the microbiome field have been very exciting. There's been a lot of work and data that's been generated, and that has really enabled the characterization of the microbiome across sites. So different body sites, but also individuals, and has shown that there's big differences between individual, but also sites. And that data has been generated because there's been massive improvement on the technology side, and that's spanning sample collection, stabilization, processing, but also a big part of those progress was due to the sequencing costs that are coming down, and also better analysis tool. And I think it's interesting to see that Diversigen has been part of those efforts. So either leading some of those studies or supporting also a lot of studies on the microbiome. And I think the field is at a step that's an exciting turning point, because I think there's been tons of link that has been established between the microbiome and human health and disease. But most of the data and the evidence that's been generated to date is based on DNA and metagenomics. And there's starting to be a body of literature that has started to look at RNA, and is kind of hinting that RNA may actually be a better analyte to look at for certain questions and studies, because it can actually give you insight into gene expression and the mechanistic and the interplay between the host and the microbiome. And that could potentially lead to discovery of new biomarker or diagnostic indicators. One of the most studied sites for the human microbiome is the gut microbiome, because it's known to play a central role in health and disease. So there's ties between the microbiome community and IBD, Crohn's disease. The gut-brain axis is also very important. But again, I think it's mostly been demonstrated through DNA-based approach. Some studies have looked at the impact of diet on the gut microbiome, and I'm highlighting just a few. So this definitely doesn't do justice to the body of literature that's out there, but people have looked at the impact of fiber intake on the gut microbiome, or also pre or probiotic. So either taking a bacteria that can colonize your gut and have beneficial effect, or taking substrate that can be specifically used by type of bacteria, and that's also going to be potentially beneficial. There's been a few studies that have also looked at different lifestyles. So for example, a vegetarian versus an omnivore diet, as well as fasting. And what's interesting is that, and it applies to most microbiome research, results are not always consistent, and they can vary depending on the cohort, the paper. And I think part of that is due that not everybody is using the same study design or tools, and sometimes they're not perfect. So that can lead to a lot of confounding factor. But also one of the questions that I think is interesting to ask is DNA the most relevant analyte to look at when you're interested in dynamic changes and the response of your gut community to a change in diet? So just a bit of background, and trying to capture here the difference between DNA and RNA, so metagenomics versus metatranscriptomics. So in my talk, when I'm going to be talking about metagenomics, this is going to refer to the microbial cells, so we're not really looking at the host. But DNA, generally speaking, is relatively easy to stabilize and extract as well. And it's really going to be useful and give you the full picture of the bacteria or microbial cells that are present in the sample. And by using metagenomic sequencing, you can also look at all the genes that are present in any given genome and kind of predict what function could basically be expressed in response to the environment, or anything that's happening in the sample. RNA, on the other end, I think as an interesting thing is that it's basically provides direct insight into the genes that are being expressed, but also the species that are expressing those genes. And as such, it's kind of gives you a more direct view into what is happening in the sample. And I think to this day, a lot of people have not been relying on metatranscriptomic for microbiome studies, because RNA is much more difficult to work with. It's very prone to degradation, but also like much harder to like stabilize. And also when you extract total RNA from a microbiome sample, up to like 98% of the total RNA is made up of ribosomal RNA, which contains very little useful information. So it doesn't tell you anything about the genes that are expressed, nor the active species. And I think that's where the partnership between DNA Genotek and Diversigen is interesting, because it brings like technology and expertise. So DNA Genotek has a lot of expertise on sample collection and DNA RNA stabilization. So this is a picture of our newly released OMNIgene GUT DNA RNA, while Diversigen recently launched a Metatranscriptomic pipeline. And I think combined together, those are the right tool to maybe take a second look at metatranscriptomics and like potentially all the data that lies into those data sets. So I think the question we ask at the beginning of this study is that with the proper tools, expertise, and know-how, can we actually leverage metatranscriptomic and better understand and dissect the impact of diet on the gut microbiome? And we chose to go after intermittent fasting because I think a lot of people throughout their lifetime are going to be observing some kind of a intermittent fasting regimen. And there's like a few studies that have shown that it does impact the gut microbiome. So again, DNA based, but it also has a lot of potential health benefit for the host. So weight loss, lower hypertension, like lower cholesterol, lower inflammation. So this is actually something that's good for anybody that follows intermittent fasting. And again, like I think there's been conflicting results in the literature, and not every study is like necessarily consistent. And we were curious to see if metatranscriptomic was going to give a different picture. So what we did is that we recruited 7 subjects, and we asked those subjects to follow a modified 5-2 intermittent fasting protocol. So this was a 2-week study, and every week people were asked to fast for like 2 to 3 days. So 5-2 is 5 days of normal diet and 2 days of fasting. The only modification we did on week 2, participants were asked for like to fast for 3 days. And then each of the participants during like the course of the study were asked to like collect a stool sample in the OMNIgene GUT DNA RNA device. So as you can see, we collected samples from like normal diet days as well as fasting days. So it's about 5 fasting representative samples and 5 normal diet day samples. And then we basically extracted the DNA and the RNA samples and characterized like taxonomy, function, CAZyme and AMR using Diversigen pipeline. And just to show you participant adherence to the study, so this is a graph that's showcasing average calorie intake for like the 7 donors. And as you can see, we see a drastic drop in calorie intake during the fasting days. So we're talking about on average about 620 calories per day, as opposed to close to 1,800 calories on average per day for the non-fasting days. So it's about a 2.8-fold drop in total calorie intake. So as I mentioned, all the samples collected as part of this study were collected in the OMNIgene GUT DNA and RNA to like mimic a real life scenario. We held those samples at room temperature for 8 to 10 days. So this is within the claim that we have for that particular device, and it can stabilize both DNA and RNA for up to 11 days at room temperature. And then we co-extracted DNA and RNA from the same extraction aliquot using QIAGEN RNA-Z Power Microbiome, just showing you the quality of the samples that we were able to recover. So here on the top right, you can see RNA and DNA, very high quality, and that's all the time points that were collected from one of the donors, so T1 through T60. And then the extracted DNA and RNA were put through the Diversigen metagenomic or metatranscriptomic pipeline. And again, we looked at taxonomy, function, CAZyme, as well as AMR. So without further ado, I'll jump into some of the results that were generated as part of this study. And one of the first things that we looked at were CAZyme. So CAZyme stands for carbohydrate active enzyme. And these is like a large group of enzymes that's involved in the metabolism of carbohydrate or modification of carbohydrate, so the digestion. And it's a large family, like there's like 5 classes and over 300 families. Those enzymes are like critical for like colonic bacteria because they're involved in all aspects of carbohydrate metabolism, and colonic bacteria tend to rely a lot on the carbohydrate from the diet, everything that's not digested by the host. Also, interestingly, I think everybody has heard about short-chain fatty acids or SFCAs, and SFCAs are like technically end byproduct of some of those carbohydrate metabolic pathways, and they've been shown to like have health benefits for the host. So when we looked at DNA versus RNA, and again, looking at the CAZyme in that case, the relative abundance, you see that the two analytes paint very different picture. So when looking at the DNA profile, you can see that generally speaking, we see like very stable abundance of those CAZyme over time and across the entire course of the 2-week study. And there's very little differences between fasting and non-fasting days. So fasting days are highlighted here in pink. And generally speaking, we did see like very good stability and very little change. And keep in mind that when we look at the DNA, we're not looking at gene expression. We're actually predicting the function that are present in the genome of the organism that are present in the sample. What I think is exciting and like shows you how like different RNA is that when you look at the RNA profile, you do see that they're like much more dynamic and variable, and you see that each of the donor is showing like pretty significant changes in the expression, because we're looking at expression here of those like CAZyme over the course of the study. And although we did not see like consistent changes that are like necessarily tied to the diet across the entire cohort, there's a couple of donors that did seem to like show diet-induced changes. So donor 117 was one of them, while other donor like showed dynamic changes through the course of the study, but not necessarily changes that are tied to like the diet. And I'll touch a bit more on that afterwards. We also looked at AMR. I just don't have time to go through that data, but we got a very similar story. So AMR stands for antimicrobial resistance gene. And when we compare DNA and RNA profile, what we did see is that DNA profiles were generally speaking very stable and didn't change much with the diet or over the course of the study, whereas RNA level did show a lot more changes in relative abundance of those genes, but it was, again, very donor specific and not necessarily tied to like fasting versus non-fasting. So we also look at taxonomy, and those plots are looking at species level beta diversity. And as you can see, again, we're comparing DNA versus the RNA profile. So DNA profile is something that a lot of data has been generated that looks like this. So you can see that generally speaking, all the samples that were collected from the same donor tend to cluster together, and this is because each of the donor has a very unique microbiome profile, and that profile is like stable over time when you're looking at DNA and metagenomic profile. We do see a couple of donors where you start to see potentially the fasting versus the non-fasting days. So fasting days are the open symbol, whereas the non-fasting days are the closed symbol. You do see like clustering of the samples per the diet, but as you can see, it's very donor dependent, and there's only 2 donors that seems to like show that pattern. That would be donor 14 and 17. Looking at the RNA provides and paints like a very different picture. Like, first of all, you do see that the ellipses and the variability that we're able to capture across all the time points that were collected for each of the sample is much bigger. So you do see there's more spread. And it's, again, very donor dependent. Some donors have more variability to begin with, whereas some donors show like pretty tight and consistent taxonomy profile. And same story, we do see a couple of donors that seems to show a response that's tied to the diet, where you start to see clustering of the sample based on like fasting versus the non-fasting days. At the start of the study, the hope that we were going to see like changes that were going to be consistent across all 7 donors. It turns out that we didn't see any consistent changes, and each donor was like showing very individualized responses. And that's best exemplified by those graphs where we're looking at functional profile over time. So donor 1112 is what we would call a non-responder. as you can see, as you track all of the samples that were collected as part of the study, you don't see a pattern that seems to line up with like the diet. So it kind of follows and goes all over the place. And there are changes that we're capturing, but they're not tied to the diet. Donor 1117 on the other end is very interesting, and that's what we would call a strong responder. You really do see clustering of the time points based on the diet. And as you can see, all the non-fasting, so there's a mistake on this slide, the non-fasting days actually cluster on the left end of the graph, whereas the fasting days cluster away. And as you follow the two fasting period, you see that they cluster together, and the day of non-fasting in between goes all the way back on the left side. So it's exciting because you really see that time dependent changes that's tied to the diet. So we also looked at differential abundance of KOs. So KOs stands for like KEGG orthologs, and based on both genomic and metatranscriptomic data. When we look at the DNA, so similar story, you do see very few of those KOs that are showing a differential abundance. So whether they're increase in abundance or like decrease. But when you look at the RNA, it gives like a very different picture. You see that the changes are like much more obvious. And you do see a couple of donors where there's very, very little change or no change at all. So those one would be the non-responder. But there's like the donor 114 to like 117 like show responses with some of the KOs increasing in relative expression and other KOs decreasing in relative expression. And again, you see a gradient across those participants. So there's a very strong responder for like donor 17, and then like a lower response for donor 16. So this is kind of the same data, but like a different graph. And this is like showing at donor 117 specifically, so a strong responder, and comparing metagenomics, so DNA, versus metatranscriptomic profiles, RNA. And as you can see, when we look at the top 100 differentially abundant function in that sample, and we compare fasting versus non-fasting days, you see that the DNA profile like do show changes over time. So each time point has differential relative abundance for some of this function, but there's no strong correlation between those changes with the diet. So you see the two first fasting days are here, and then the next three are here, and it doesn't seem to like give any kind of particular pattern. However, when you start to look at the RNA profile, what's very interesting is that you see function that basically turn on and off based on the diet. So everything that's on the left side here, you see is green. So it's expressed more in the non-fasting days and very little expression during the non-fasting days, but you see that the pattern completely changed when you go through that fasting period. And then you go back to your normal diet, it shifts again, and those functions turn on and off. And same thing during the second intermittent fasting. So I think it's very exciting because RNA like is really able to like show consistent functional changes for that donor, but it also shows changes that you cannot see with DNA-based approach. So we did a deeper dive. As I mentioned, we were not able to identify consistent changes that were seen across all of the participants in response to the diet. So what we started to look at were like individual changes within each of the participants, and we like looked at differential abundance of specific KOs in fasting versus non-fasting days. And so there's some function that were downregulated. So this is all for donor 117, but we saw a similar picture for the other responder and KOs that were upregulated. And as you can see, one of the patterns that we do see looking at the data is that there seems to be overrepresentation of amino acid and protein metabolic pathways. So for example, biosynthesis of amino acid is upregulated in 6 species, but also downregulated in like approximately like 20 to 25 species. So that shows you that bacteria, in response to the diet, some of them shut down biosynthesis of amino acid, while other like jumpstart amino acid synthesis. One very consistent change that we did see, generally speaking, is most of the bacterial species were shutting down ribosomal function. So you see upward of 60 species did show downregulation of ribosomal. And generally speaking, we did see more downregulation, and a lot of the downregulated pathway were around amino acid synthesis and metabolic pathways. So it's very interesting because we do see like global changes, but we're also able to like pick up species levels changes, and different species respond differently to the diet. So now I think we have to like go back to the biological question that we were asking, and what is the true impact of the intermittent fasting on the gut microbiome? And I think generally speaking, we tend to approach nutrition from the host. So what the host eats and what impact it's going to have on him. But I think what we kind of didn't necessarily consider at the start of this study is like the diet is also going to be impacting the gut microbiome, because what you eat eventually makes it all the way to the colon. And the main nutrients that are available in the distal gut are actually undigested fiber and carbohydrate. So there's carbohydrate that the host, the human, can process, but there's some that they cannot, and they're usually like substrate that the bacteria use to like get carbon and energy from. And a second source of energy and a nutrient for like colonic bacteria is unabsorbed amino acid and protein. And when we look back at the data and looked at micronutrient intake during the intervention study and that fasting, what was very striking is that we did see most of the macronutrient like being impacted by the fasting days. So you see carbohydrate go down by a factor of 2, but the most impacted were actually fat and protein, and like fiber and carbs were like not impacted as much. And what's interesting is that based on what we know, colonic bacteria utilize for the metabolism, what we think is happening, and our like current hypothesis is that the fasting severely restricted the bioavailability of nitrogen and amino acid in the distal gut, and that's what basically trickled and caused a decrease in like protein synthesis pathways and the changes in the amino synthesis pathways. And it actually makes sense because there's a body of literature that actually shows that nitrogen is a rate-limiting nutrient in the distal gut, and that the colonic bacteria are like competing for like the nitrogen and the amino acids that are present in there. So it seems that the biology of it all is emerging from our data, which is very exciting. Just as a summary, as a conclusion, so I think intermittent fasting, and we were able to show that it induces very highly personalized gut microbiome responses. We do see, generally speaking, similar taxonomic and functional features in the DNA and the RNA. But I think what's interesting is that the changes that we see in the RNA are more dynamic, and they're far more drastic than anything that's picked up with DNA-based approach. And that applies to like function, CAZyme, but also the antimicrobial genes. And as such, I think MetaT like truly captures pattern that MGS does not detect. And I think that's the best illustration we have of this, where you really see the RNA like lighting up based on the diet and the fasting. We saw significant heterogeneity in response to like the diet and the fasting. I think ultimately it's rooted in the unique communities that's present within each of the donor. So those are like biological difference, and I think each donor is going to respond differently to like fasting. But I think there's other confounding factors that we've now are like trying to consider. Transit time is a big one. I think as part of this study, we were collecting like samples, and we were thinking that every sample that was collected was representative of the day before, but that's actually not necessarily true, because it can be like a wide range of transit time, and maybe some of the samples are not actually representative of fasting days. And we're obviously, so this is still very preliminary, we're still continuing to explore this data in a multi-omic and precision medicine context, and trying to like really get at what's driving the individual response to like fasting and diet, and which marker can potentially help predict that response. So as I mentioned, a lot of people do intermittent fasting, including the 5-2 protocol, and it would be great to like be able to like predict the response of any given individual based on its microbiome, and like tell them if it's actually something that will be beneficial for them. And I think we're still trying to like tease out if MetaT can provide better insight than MGS, but our preliminary data seem to suggest so. And yeah, like, thank you very much for your attention. I'll be happy to take any question at this point, or like, if you want to like come chat with us at the booth later, like, please do so. Thank you very much.